This is the second of two videos which looks at factors that affect the activity of enzymes. In the first video we looked at the effect of substrate concentration on enzyme activity. In this we focus on the effect of temperature, pH and inhibitors. So, a quick reminder of the key features of the structures of enzymes that are relevant to this topic. So here we can see part of the tertiary structure of an enzyme, the complex folding of the polypeptide chain caused by interactions between amino acid side groups, such as hydrogen bonds and ionic bonds here. That complex folding, of course, creates the overall complex shape of an enzyme, and in particular the active site, the cleft into which the substrate will fit and bind. And here's a diagram showing how that folding creates a binding site and how the amino acid side chains that line the binding site are able to bond to this particular substrate here, making use of interactions such as hydrogen bonds again and ionic interactions. So, let's have a look at the first of those factors, the effect of temperature. You'll be familiar with the idea, probably from at least GCSE, that enzymes have an optimum temperature at which the rate of reaction is a maximum. And so we have a graph here in which, on the left-hand side, the behaviour of the enzyme matches that of most chemical substances, i.e. that as you increase the rate of reaction, as you increase the temperature of a reaction mixture, the rate of reaction increases. Now we need to be able to explain that, and again you can explain that using completely conventional chemistry. As you're increasing the temperature, you're giving the molecules, the substrate and enzyme molecules, more kinetic energy, and consequently when collisions between those molecules occur, there's a greater chance that those collisions will be successful. In other words, that the molecules will possess sufficient energy to overcome the activation energy that prevents reactions from occurring. So far, that's extremely familiar. Then we have to consider what happens on the other side of the graph when we get beyond the optimum temperature. And this feature is specific really to enzymes. What's happening here is that as you increase temperature, the rate of reaction decreases really quite dramatically. Why should that be? Well, it's due to a process known as enzyme denaturing. Now, when you're answering questions on this, it's rarely enough simply to state the enzyme is denatured. What you will need to do is to be able to explain what that means in terms of enzyme structure. And in the next slide, we'll look at that. Okay, so here's denaturing occurring. As you can see, what's happening here is that we're breaking down the complex tertiary structure, and indeed the secondary structures as well, to end up with an enzyme that doesn't have any kind of complex overall shape. So we lose that tertiary structure. In particular, we lose the active site, the cleft of a particular shape, which plays such an important role in enzyme catalysis. So denaturing is the breaking of the weak bonds that usually hold the tertiary structure and indeed secondary structure in place. Those weak bonds are often, of course, hydrogen bonds. Once you break those bonds, there's nothing holding the structure in place, and the polypeptide chain is able to take up any conformation that happens to randomly occur, and so we lose the specific shape of the enzyme. Because of that, and because particularly the active site is altered in shape, the substrate molecule can no longer bind to the active site, hence the enzyme catalyzed mechanism can no longer occur. Very important whenever you're discussing enzyme activity to mention active site and the shape of the active site in particular. Now we can look at the effect of pH. It's often believed that enzymes have an optimum pH that's always at 7. Well, that isn't true. They certainly do have an optimum pH at which they work most effectively, but it isn't always 7. Here we can see, for example, an enzyme that works in the stomach of the human body, pepsin, and as you might expect, it works best under acidic conditions. So the key thing to realise is that as you go away from the optimum pH, even by 1 or 2 pH units, the activity changes really quite dramatically. The activity drops the further away you get from the optimum pH, both as you increase the pH and as you decrease the pH. And students often believe that this is due to the same kind of denaturing process that occurs when you change temperature, when you increase temperature beyond the optimum. Well, it's not quite as simple as that. Certainly, if you increase pH dramatically, you may well end up with an enzyme denaturing. But the subtle effect that occurs for small pH changes will need a more sophisticated explanation. So what is that explanation? Well, we talked earlier on about the fact that there were ionic interactions that helped to maintain the tertiary structure and indeed helped to hold the substrate in place by bonding ionically to amino acid side chain. It's worth being clear, I think, which amino acid side groups are particularly affected by pH, and these are the ones that are involved in that ionic bonding, so they're ones that contain NH2 groups or carboxylic acid groups. Of course, you'll notice here that in the particular pH in which you find this particular enzyme, there are certain charge distributions on those groups, and it's those charge distributions, of course, that allow the ionic bond to form. So, if you change the pH, you change the H plus or OH minus concentration, what happens is that the charges on these sorts of groups are going to change. So, NH2 groups and COH groups may gain or lose charges as you change pH. Well, once you change the charges, you change the possibility for ionic bonding here and here, and so either the active site shape changes or its ability to form bonds to the substrate changes. Either way, you will lose enzyme activity. So that's a much more subtle effect than simply denaturing. And finally, we come to inhibitors. This is quite a complex topic in biology, but in chemistry we can treat it much more simply. You simply need to be aware that inhibitors are substances, molecules, that will either block the active site or alter its structure in some way so that substrate molecules cannot bind. The blocking, you can see, is relatively straightforward to explain. What often happens is the inhibitor has quite a similar shape to the structure of the substrate, and consequently it can fit into at least part of the active site, but when it's there, it doesn't bond quite so well, and doesn't react, and just stays there. And as a result, the substrate is unable to fit into the active site while the competitive inhibitor is in place. There's a second kind of inhibitor which actually bonds to a second active site, so it has a very different shape to the substrate, and the energy that's released when the bonds form to that second active site can be enough to slightly alter the shape of the active site itself and mean that the substrate can no longer fit in and can no longer bind. This often is called non-competitive inhibition. You don't need to be aware of the details of that, simply know that inhibitors can operate in this way. Examples of inhibitors, a lot of transition metal ions, particularly heavy metal ions like mercury ions, are able to bond to the active site here, and that explains one reason why heavy metal ions are often extremely toxic. They'll affect the functioning of enzymes within the human body. That ends this second video looking at the factors that affect enzyme activity.